also confirm if the audio is clear to you. I'll also share the screen now. I hope the screen is visible. So if the audio is clear and the screen is visible, can you please confirm that in the chat box? I'm audible, right? Or is there any issue with audio? Janani and Mahasinu, is the audio clear from my end? And also, is the screen visible? Okay, thank you so much. So yes, with that we'll start with the session today. If you have any doubts regarding the previous sessions or lectures, you can just post in the chat box. I'll try to address them, and you can also unmute yourself as well in order to ask the doubts. So in this week's we'll see some more techniques in environmental microbiology and some concepts about bioremediation. As in the previous weeks, we'll solve some questions, and then from that we'll discuss what the concept is about, and as usual that we have done for all the previous weeks, we'll do it this week also. So the first question for today is, algae can be found in dash zone in a Vinogradsky column. Anyone wants to try in which zone in a Vinogradsky column can you find algae? Are you all aware what is a Vinogradsky column is? or the aerobic zone. Okay, so this is typically a Vinogradsky column. So what you do in a Vinogradsky column is you try to mimic a ecosystem. One sec. So in a Vinogradsky column, you try to mimic or create a miniature ecosystem, and you study different chemical gradients here from top to bottom. So the first or the top layer is going to be the aerobic zone or the oxic zone, and next you will have the microaerophilic zone, and finally you will have the anaerobic zone. Okay, so it has a oxygen gradient. So because the top layer will be exposed to oxygen, there is aerobic zone, whereas the dissolution of oxygen is not possible inside the column, and henceforth your bottom is going to be anaerobic zone, as you can see in this image. So in the anaerobic zone also you will have different layers, where you have a layer where organisms consuming organic acids will be present, followed by the organisms that consume hydrogen sulfide in a moderate concentration, and then in a higher concentration. All of these will be present, like as you can see. So and also the apart from this, so, oxygen gradient, that also simultaneously creates a redox gradient as well, because the terminal electron acceptor for the organism is also going to change. So therefore algae is a photosynthetic organism basically, so you will find it in the top layer. I hope it is clear. So this is the second question. When there is a chance of product inhibition, sequential batch reactor is preferred. Is this statement true or false? Okay. First of all, are you aware what is product inhibition? So whenever there is a process, there is some substrate that is being consumed by a microorganism and a product is formed. Okay, imagine this like a chemical reaction. So you give products, and you give the substrate, and you get a product. A plus B gives C plus D is a simple equation all of us would have learned in basic chemistry, right? So here, and the medium by which the reaction happens is through microorganisms. Okay, and we all know that there is some thermodynamic and kinetics involved in proceeding of a forward reaction where the production of the product from the reactant is going to like get slowed down, uh, because there is more amount of product. So when we talk about biochemical kinetics, sometimes enzymes are inhibited, enzyme activity is inhibited even by the accumulation of product in the reactor system. Okay, this process where the formation of product and the high concentration of product slows down the forward reaction is known as the product inhibition. So this is one of the concerns in bioremediation. So let's when we talk about something, bioremediation or a biological waste treatment process, we are going to add some waste material and we are trying to convert it into some other form using microorganisms. So for example, when we talk about an anaerobic digestion process, you feed in your organic waste, the microbes consume it, and finally you get methane as the product, or a hydrogen as the product, or volatile fatty acids as the product. So when there is a large high concentration or production of these products can itself further inhibit the proceeding of the forward reaction. Okay, so this concept is product inhibition, and obviously from a engineering perspective or in applied perspectives, we will not be interested in product inhibitions occurring. So we need to find ways to um, overcome this product inhibition. So here they are telling that sequential batch reactor can potentially help us overcome this product inhibition. So before that, you know what a batch reactor is, right? So a batch reactor, in the sense, you have a reactor, you feed in your substrate, set up the reaction, and let the reaction get over. You neither add more reactant into the system while the reaction is in process, nor do you remove anything, any product or any reaction mixture from the system while the reaction is running. So you allow the reaction to get completed. So it starts, it reacts, and then it completes, and finally after the reaction is completed, you take out your products or the reaction mixture out. So this is how a typical batch reactor works. In contrast, in a continuous reactor, what you do, you continuously feed in the substrate and continuously remove the products in the system. Okay, so in a sequential batch is kind of a middle ground where you have more than one reactor. Let's say some 2 or 4 reactors. So first what happens, you feed in the substrate into one chamber, the reaction starts, proceeds, and the reaction also completes. Now what you do, you transfer this to the next chamber where no reaction is happening, but you are trying to settle the particles. For example, in any aerobic or anaerobic process, you try to send it to another system where you do not want the microbe to grow and all, you just want the solid liquid separation. Like the sludge part alone usually gets settled, and the liquid part just stays at the bottom. This is basically a density based or weight based settling separation kind of stuff. In the meantime, in the first reactor, again what you do, you add in a new set of reactants, substrate and the inoculum, and once the settling has happened, you then transfer it from the second to the third now, where you try to separate the solid and liquid, and finally you try to clear off the entire reactor. Okay, so the typical steps include, you fill, you allow for the reaction, you allow for the settling, and then you finally decant. So these are the 4 major steps in a sequential batch reactor. So you allow the process to happen in sequence, but it happens only in batch. It's not a continuous process. Okay. So now based on these 2 information, I'll just wait for a couple of more minutes for you to think and answer whether sequential batch the usage of sequential batch reactor can help us overcome product inhibition, or will it be the reason where product inhibition is going to be the reason for affecting our process. So you have to answer, when there is a chance of product inhibition, sequential batch reactor is preferred. Whether sequential batch reactor is useful to overcome the limitations of product inhibition. True or false? initial phase, when before starting the reaction, you have the liberty to have low product concentration. Only then the reactant will be more. On the onset of the experiment, it's a batch experiment, so you are neither adding anything or taking anything out. So if you add new reactant, then you are diluting the concentration of product in your reactor. Okay, in that case also product inhibition will not be like much. So this happens in a fed batch or semi batch reactors. So in semi batch reactors, what you typically do is you don't take any product out, but you continuously keep adding some substrate. In that case, what you do is you try to dilute the entire reactor. When the product concentration is more and you add more reactant, then the product concentration gets diluted. This doesn't happen in a sequential batch reactor. Neither do you remove anything from the sequential batch reactor. So still the product concentration stays high only in that reactor. So in that case, the chance of product inhibition is more when you use a sequential batch reactor. Therefore, it is not preferred. I hope that was clear. Okay, so to enrich green sulfur bacteria, we incubate it in dash condition with dash as electron donor. well. The kind of photosynthesis that happens is anoxygenic photosynthesis. So this process that does not even liberate any oxygen. So it is anoxygenic photosynthesis, and the conditions required are anoxic or anaerobic conditions, and the electron donor should be hydrogen sulfide. So since we saw about this Vinogradsky column as well, usually this is present in the very bottom of the Vinogradsky column where there is no oxygen. nitrogen directly as the nitrogen source. So before using nitrogen directly, all these bacteria should have the capability to fix nitrogen. So cyanobacteria, they are highly efficient in fixing nitrogen. So maybe commonly we so make sure that cyanobacteria we choose as answer for this question, but also the other options are right as well, though these names may be not so common. So in cyanobacteria they use specialized cells uh, called heterocysts, in which they protect this nitrogenase enzyme. So nitrogenase enzyme is generally very sensitive to oxygen, so it's inhibited by oxygen. So in that case, cyanobacteria have heterocysts where they can protect this nitrogenase enzyme, and also they have this capability to fix nitrogen. Similarly, in heliobacteria, this is also an anaerobic bacteria and it is phototrophic. Okay, they also have this nitrogen fixation capability, and in purple non-sulfur bacteria they can fix nitrogen under specific conditions, not always but under specific conditions they can also fix nitrogen. And usually when this is phototrophic and when there is no other nitrogen source available, the bacteria tends to use nitrogen itself as direct nitrogen source, and only that time it exploits its ability to fix nitrogen. So the next question is, for enriching dark chemolithotropic bacteria, the medium must lack what? Exactly. So we need chemolithotropic bacteria. When we give organic matter, the organism tends to use the organic matter which contains carbon readily. So instead of that, you have to supply it with inorganic, readily available inorganic compounds, so that we try to enrich only the lithotropes. So without organic matter, the other organisms will eventually be eliminated from the system, and only lithotropes can survive, and that's how we enrich them. Okay. The electron donor for Thiobacillus species is what? to autotroph. So obviously it needs inorganic compounds, and especially because it is thiobacillus, sulfur is primarily involved. So hydrogen sulfide, elemental sulfur, thiosulfate, as well as sulfite can also be used as electron donor. And at the end of this process, these compounds are oxidized into sulfuric acid or sulfate compounds. So primarily you need inorganic compounds because this is a chemolithoautotroph, and because it's thiobacillus you are using sulfur rich compounds like hydrogen sulfide, elemental sulfur, thiosulfate, and sulfite also sometimes. Okay, I guess most of you are from microbiology and stuff only, so this also is one more easy question. Autoclaving is carried out at which temperature? Yeah, it is 121 degrees Celsius at 15 psi pressure exactly. Uh, so, why do we use this specific temperature and pressure? And one more factor is time as well, which is around 15 minutes. So, autoclaving is a sterilization technique wherein we try to eliminate other any microorganisms that is present in our reaction media, reaction vessels, or any other stuff that we use in microbiological systems. So this is basically a sterilization technique, and there are also different sterilization techniques like heat based, which again is autoclave, or there is like direct or dry heating in a hot air oven, that is also possible. We use flame, like inside the continuous heat is the major driving factor that brings about the sterilization, and that is why we use a specific temperature and pressure, and based on that there is also a time set. If you are going to set this amount of temperature and pressure, and within this duration uh, the microorganisms will be dead. And of course, as you rightly mentioned, we try to destroy even the heat resistant microbes. And one more thing is, we try to destroy the spores, not only the microorganisms, we try to destroy their spores as well. So when we try to expose the microbe to any adverse condition, what happens is that microbe tends to form spores, and there are already spores that could be present in the medium or in the glass vessels. So when we set this temperature to 121 degrees Celsius, not ratherly at some 100 degrees Celsius or something, even the spores are destroyed in the process. And one more thing is that initially the temperature calculations and all were made in Fahrenheit system, where this is like uh, 250 degree Fahrenheit, and when you convert it comes to around 121.1 or 121.5 degrees Celsius, something like that. But for in biochemical terms, this is kind of a sweet spot that also kills the spores along with the microorganisms that are alive. Okay, in a plate count method, the CFU must be between what range? To consider that plate for counting and to identify the number of microorganisms in the sample, what should be the range? 30 to 300 is suggested by uh, USDA and AOAC standards, which is generally used in microbiology, and 25 to 250 colonies is generally suggested by uh, APHA or FDA standards. And in when we talk about ISO, it suggests 15 to 300. So obviously 300 could be the upper limit, and your bottom limit differs between 15, 25, and 30. So I have just used this USDA AOAC standards, and my answer is just 30 to 300, and 25 to 250 is also right in, and it depends on the context in which we talk. So when the colony count is less than 30, what happens is it could be because of chance. It's like too few to count. We have two concepts. One is too numerous to count or too many to count, and other one is the too few to count. Okay, so here what happens is when the number of colonies that has grown in the plate is less than 30, even a single error will give us a very high error margin. So in that case, the reliability of the data itself is questioned. Okay, so let's say there is one colony that did not grow by chance, or there was no enough incubation time, something or the other happened. The non-growth of that one colony is going to affect the entire calculation to a very greater extent. So that is why we have a lower limit of 30 or 25 colony forming units, and the upper limit is usually 300, because if it is more than 300, the plate will be overcrowded, and the overcrowding itself will inhibit the growth. Like there will be a very high competition for the nutrients, the growth will be inhibited, and also it will make the colonies overlap. So we need to see individual colonies and count them. So sometimes because there is overcrowd, maybe two colonies grow together and we still cannot distinguish them very distinctly. So that also happens, and that is when you call it as too numerous to count. So ideally the plate should have the number of colonies between 30 to 300, and then you call that plate valid. So that is why whenever you have a sample, you try to make different dilutions of it, and you only take up the plates which have the count between 30 to 300. What is the indicator that is used in anaerobic culture? Exactly, we use methylene blue, and as well as we also use these as well. microorganisms that don't use oxygen as terminal electron acceptor, but some other compounds as their terminal electron acceptors. So here we have different organisms called obligate anaerobes, facultative anaerobes, and okay, and microaerophiles, whatever. So when we talk about this obligate anaerobes, these are anaerobes compulsorily require anaerobic condition. If there is even small amount of oxygen, it's very lethal to the growth of these microorganisms. And when you study anaerobic processes, you try to strictly maintain anaerobic conditions in the process. So in order to monitor whether there is strict anaerobic condition, you use these indicators, and these indicators are basically redox indicators. So based on the redox potential, they can change color. Based on the redox potential, in the sense based on the presence of oxygen or absence of oxygen, they can change color. So when we talk about methylene blue, in the presence of oxygen, methylene blue is oxidized and it remains blue in color, whereas in the absence of oxygen, it is in a reduced form, generally called as leucomethylene blue, and this form is colorless form. So usually it should stay colorless. If it turns blue, then we can assume that there is some oxygen intrusion into the system. When we talk about risazurin, it is also a redox indicator. In oxidized state, it is pink in color, whereas in the reduced state, in the absence of oxygen, it is colorless. Out of the following techniques, which technique is not used to count microbes? Okay, so the answer here is serial dilution. We'll see about each of the technique given in the options. So first, when we talk about serial dilution, this is kind of a preparatory process that we do before plate count. So in by serial dilution we cannot get any numbers or values of how many microbes are present in a particular sample. Okay, so what happens is when we take an environmental sample, we don't know how many number of microorganisms are going to be present in the sample. And a few questions back we talked about the optimum CFU that we have to consider. So in that we found that a maximum of 300 colonies CFUs in a plate is allowed. More than that, it is too numerous to count, and we omit that. And in environmental samples are rich in microorganisms, so obviously there will be more than 300. So we try to dilute it. And how we do a serial dilution is that we take up the sorry, there was a audio crash. Okay, so in serial dilution we take up the sample and we have some buffer, saline buffer or autoclaved water, whatever prepared. So to we usually perform it in 10 times dilution, which means we take 9 ml of these buffers or water and add 1 ml of our sample, and from this we transfer 1 ml to the next tube containing another 9 ml of buffer or water. So this sequential addition of 1 ml into 9 ml dilutes the sample 10 times each time. So when you do it twice, you have diluted it 100 times. When you do it thrice, you have diluted it 1000 times. So that is how you mark it as 10 to the power minus 1, 10 to the power minus 2, and 10 to the power minus 3 dilutions, right? And then finally, when you have the plate count value, you again multiply the dilution factor. So when you have found some 30 colonies with 3 times thrice of 10 times dilution, you multiply 1000 and say there were 30,000 cells in the initial sample. So this is how you work with serial dilution process. But after serial dilution process, you should work with plate count or agar plating method, and then only you can find the number of microbes. Whereas in microscopy, yeah, you can use a hemocytometer or other chambers where you can directly count the number of cells that are present in the sample. And plate count, we again discussed it with serial dilution itself. We have a Petri plate, we inoculate it, and then find the number of cells. In microscopy, maybe you have some staining techniques in which you can differentiate between live and dead cells, but generally you don't differentiate between live and dead cells. Whereas in plate count, viable cells can only grow. There is no space dead cells can grow in plate count method. So that is a little bias between these two methods. When we talk about a qPCR method, PCR we know it's a polymerase chain reaction where we have DNA molecules, okay, and we try to amplify the DNA. So what we typically do is we have the DNA and we try to add some tags to it, fluorescent tags or other dye-based tags or intercalating tags, and we perform the PCR. Once there is ample amount of DNA that has been multiplied or replicated, then the fluorescent signal or the dye signal tends to be detected above the background or noise. And now we know what is the concentration. Based on the signal, we can know what is the concentration of DNA, and we can backtrack the concentration of DNA and then trace it back to the amount of microorganisms or the number of microorganisms that is present. Very indirect way, but still you can count microbes using a quantitative PCR as well. Serial dilution is the only process where only with that you cannot count the number of microbes. Okay, another true or false question. When GC bonds are more, DNA does not travel farther in DGG. So for this, you have to know what a DGG is. I'll give you some time to think, and then after that I'll tell you what a DGG is. also has different denaturants and in different concentrations. The denaturants usually include urea and formamide. So the key mechanism here is when a DNA is exposed to this denaturants, they try to unwind, like the two strands try to separate, and the DNA fragments travel through the gel until they reach a concentration of denaturant that causes the double stranded DNA to melt into single strands, and after this the halt halts this process halts. Okay, so what happens here when we have a GC rich sequence? Okay, so when there is a GC rich sequence, we know that the GC bond is a triple bond, whereas the AT bond is a double bond. So this triple bond is very difficult to break. When a DNA sequence, two DNA sequences are of similar length, let's say, but one has a higher GC bond and the other doesn't have that much of GC bonds. So breaking of the DNA which has high number of GC bonds is very difficult. So it travels farther. Only when this denaturation is so fast and the denaturation happens so quickly, both the DNA starts to move. When the denaturation rate is very higher, it easily gets denatured and then stops. The movement of the DNA through the gel actually stops. Whereas when the GC bonds are high, the denaturation rate is very low. So what happens? The DNA tends to move through the gel for a higher or a longer distance. So when GC bonds are more, DNA travels farther in DGT. So that should be the statement, and that is why this is false. I hope this is clear. Are there any doubts? Okay, bioaugmentation and biostimulation. So when we talk about bioaugmentation, we are going to augment or add microorganisms to the system. So directly we are adding something biological. Augmentation means adding, and we are adding something biological. So that is what is bioaugmentation. What could we pretty much add? Microorganisms, because they are the heroes of our biological processes, which are going to drive the entire process and the system. So when we talk about bioaugmentation, we are going to add some specialized microorganisms. Let's say we already have a consortia, but we are particularly interested. The consortia can perform a variety of reactions, but we are particularly concerned about one but aspect of the function of the consortia, like in utilization of a specific compound or production of a specific product, we are concerned about. So in that case, we try to enrich the consortium inoculum with specialized microorganisms that can help perform the targeted reaction. So this process is called a bioaugmentation process. And when we talk about biostimulation, here we try to add chemicals. We are trying to stimulate the already existing microorganism in the system or the consortia. So in order to stimulate them, like pretty much analogical to what we do in an enrichment culture. So we try to add some specialized media, a specialized nutrient to the media, which supports the growth of a particular type of microorganism. So similar to that, we are trying to add some nutrients, surfactants, electron acceptors, donors, or any other chemical component to stimulate the system to perform a particular reaction. Pretty much we will also kind of enrich the microorganisms by adding a particular kind of nutrient, because the growth of that microorganisms will be supported by what we are adding. So in bioaugmentation, you directly are going to supplement it with a microorganism itself, whereas in stimulation you are going to add a chemical compound and in turn get the microorganism do the job. So these are the two methods of accelerating a bioremediation process. So be careful and aware of the differences between both of them. Oh, another easy question regarding the process. We have come to the process aspect pretty much. The conversion of a contaminant into CO2 and H2O is called what? Name the process. Exactly, it is mineralization process. So in mineralization, there is complete degradation of the contaminant, and finally you only have the carbon dioxide and water produced from the system. So any contaminant degradation through chemical or biological methods will undergo a series of steps. It's not like in a single step process you convert the contaminant into some CO2 and H2O. There will be series of steps and you will have a number of intermediates. These intermediates can be less toxic than the contaminant itself, or in some cases they can be more toxic or detrimental than the original contaminant, these intermediate byproducts. So during the conversion of contaminants, it is essential that you ensure that finally it produces CO2 and H2O, and this entire process you call it as mineralization process, which is complete degradation. Okay. When there is a change in redox state or conversion to an intermediate metabolite that may differ in toxicity or mobility, what do we call that phenomenon? Transformation or biotransformation in particular is the process in which we have a contaminant and we have a chemical change in the redox state of the contaminant. This process is called transformation. Any compounds that are not occurring within the normal metabolic pathways of a biological system are known as for the microorganism. When you have more than one carbon source, you have a lag phase, log phase, again a lag phase, log phase, and then it goes to stationary and then decline. So this is a typical representation. So if that graph is given to you, what do you call it? So the answer for this is dioxy growth. As I told you, when you imagine a growth curve. Okay. So typically what you have is a lag, log, stationary and a decline. Right? But when there are more than one carbon source present in the system, the microbe naturally tends to use the first carbon, like easily utilizable carbon source first. Let's say we have glucose and we also have cellulose. Okay, both are carbon sources. So when we have both of these, the microbe naturally tends to have glucose first because it is easily utilizable. So for this you have a lag phase and then the microbe enters the log phase. After that it should ideally go to a stationary phase. But you still have nutrient left in the system which is the cellulose. So what happens is it again takes a little amount of a lag phase to get adapted to cellulose, like to synthesize cellulose, break down and stuff. And then after that it again goes to a log phase, like it just increases. And after the utilization of cellulose also it goes to a stationary phase. So what what you can typically imagine is, so this is your growth curve. So this is a lag phase, this is a log phase, and here a glucose is used. After that it should go to stationary phase. No, it won't go. What it does is it again goes to a little lag and then again increases to log. There it tries to use the cellulose, and then it goes to the stationary phase and then to the decline. This is how your typical growth curve looks for a dioxic growth. When you find this pattern you call it as dioxic growth. Okay, so which of the following statements about environmental remediation of aromatic compounds is true? Exactly, D is the right answer. So aromatic compounds have the cyclic ring structures, and they are very stable and difficult to degrade. So they cannot be easily degraded by microorganisms, nor they can only be degraded by chemical methods. They can be degraded by microorganisms, but yeah, they are very recalcitrant, and this is a very slow process. The kinetics is very slow, and the microbe tends to struggle a lot to bring about this biodegradation. And also, chemical oxidation methods work well for aromatic compounds, but they are not the only ways to treat aromatic contamination. And physical methods are generally not very effective in treating aromatic compounds. Yeah, we have um, thermal methods like incineration and all, but they can cause secondary pollution, and they are not so very effective in treatment of aromatic compounds. The reason for all the other three statements to be true is the fact that aromatic compounds are recalcitrant and are highly stable and also very highly toxic. Polyhydroxybutyrate is a pollutant. So, which of the following statements about the degradation of PHB is true? Is an important process for bioremediation of plastic waste. It's not that only bacteria can degrade PHPs, and of course, when there is microbial degradation, they require enzymes. It's not that they don't require enzymes, and they also produce other products. Like when this happens in anaerobic environment, methane is produced. So, degradation process does not only produce CO2 and water. Again, a true or false question. The substrates involved in co-metabolism are growth associated. Obviously, it is false. So, co-metabolism is a concept wherein two substrates are present and both of them are utilized by the microorganism, in which one of the substrate is growth associated while the other substrate is a non-growth associated one. So that is called a co-metabolism. Okay, like in the growth associated substrate is the primary substrate, and that acts as the main source of carbon and energy for the microorganism. Whereas this um, non-growth associated substrate is a secondary substrate, and this also gets utilized by the microorganism and henceforth degraded. This is usually the pollutant or the contaminant that we can try to target. This is happening just by chance. The microorganism does not intend exactly to utilize it. When it is utilizing the primary substrate as carbon and energy source, it could also use the other substrates that are present, other components that are present there as substrate as well, though they are not contributing contributing to the growth of the microorganism directly. So, whenever we talk about co-metabolism, one is the intended metabolic process, and the other is happening just by chance. And that pretty much says that out of the two substrates, only one is growth associated, whereas the other is non-growth associated. So, transformation of co-metabolite substrates produce toxicity. Whether this statement is true or false. Transformation of co-metabolic substrate produces toxicity because that is obviously non-growth associated, and whatever metabolite that comes out of it could be detrimental to the microorganism itself. This is some context about whatever we have discussed so far. The next question is, during enrichment culture for nitrifying bacteria, why is organic carbon source excluded from the medium? try to find the molecular fingerprint. We saw how a DTG works, denaturing radiangle electrophoresis. In shotgun metagenomics, you try to identify, you basically do a shotgun sequencing where you fragment the DNA into short pieces, and then you try to identify the functional genes. So based on this, you can again map to what organisms could be present. Illumina is a high throughput sequencing method, and whole genome sequencing, obviously you're doing it to find the organisms up to a resolution of species level also. So yes, I have highlighted the wrong answer here. While uploading the PPT, I will just correct. So in a serial batch enrichment with repeated transfer, the dominant organisms are those that have which of the property? This is the last question for today. We'll just discuss this and so the right answer here is B. Survive starvation between transfers. So I think you are familiar with this concept of case strategists and R strategists. So R strategists, uh, they are they thrive in stable low nutrient environments. Okay, where there is low dilution rate. Whereas in serial batch, what happens is it favors R strategists, which are the fast growing organisms. So yeah, surviving in between uh, transfers is required, but that doesn't solve the purpose of enrichment. And also, spore production is a mechanism for extreme conditions, and that doesn't relate towards the serial batch. With that, we will close the session for today. If you have any doubts, please post in the chat box, or else thank you for joining in. Hope to see you in the next session as well. Please let me know if there is any doubt. Thank you so much for joining, and thank you for joining in. If there are any doubts, please post in the chat box.